So there are a number of points here that I want to make in terms of compare and contrast between E. coli. The first and most obvious one is that in contrast to E. coli, where cells that turn out to be persisters after drug exposure are already in a very slow or non-dividing state prior to the addition of drug, this simply isn't true in mycobacteria. So in fact, if we quantify the growth rate prior to drug addition of cells that die over here versus cells that survive over here in a particular experiment, these are data from one experiment, we find that there's absolutely no difference in the average growth rate or in the spread of growth rates between these two populations. So Bigger's hypothesis apparently is true for E. coli responding to ampicillin, but it is not true for mycobacteria responding to isoniazid. We do not at this point know whether this is a difference in the species that we're looking at or a difference in the drugs that we're looking at. This remains a challenge for the future. So that's the first take-home lesson from this sort of compare and contrast, that in E. coli, persisters are rare, slow-growing cells, and they exist prior to antibiotic exposure, whereas in Mycobacterium, the persisters are growing and dividing at completely normal rates prior to antibiotic exposure, so it's a very different phenomenon. The second point of contrast is that in E. coli, the non-persisters, those cells that actually die in the presence of the drug, continue to grow at nearly normal rates after exposure to the antibiotic. So the antibiotic doesn't stop them at all. They keep on elongating until they lyse. And that cell lysis occurs actually quite rapidly after exposure to the drug. In contrast, in mycobacteria, the non-persisters, that is the bulk of the population, arrest growth really within minutes after exposure to this drug, isoniazid. So it's a very rapid response to the drug. But then cell lysis begins only after quite a prolonged delay of about 24 hours or so after initial exposure to the drug. So it's a completely different, really opposite response to drug exposure. In E. coli, after washout of the drug in the microfluidic device, the persisters do not, in the continued presence of the drug, the persisters do not resume growth as long as we continue to pump it through the microfluidic device. In fact, they do not resume growth until after washout of the drug, whereas in Mycobacterium, the persisters, as you saw, resume growth and division after prolonged, that is, many days of antibiotic exposure, although some of the progeny of those divisions, in fact, do not survive. And when we wash out the drug in the microfluidic device, we find that E. coli persisters reactivate with slow and stochastic kinetics after antibiotic washout. So it's almost as though it's a process of radioactive decay determining whether a cell can reactivate at a given time or not after drug washout. Whereas in mycobacteria, those persisters reactivate almost immediately, very rapidly after drug washout and with uniform kinetics. So it seems very clear that the drug is actually keeping them in this non-dividing state. As soon as that pressure is removed, the bacteria start to grow again.